Okay, part 6 of chapter 3, lactic acid. This is a really important section of this chapter. And for some of you, it may be a presentation of a different viewpoint of lactic acid. Historically, we have thought lactic acid is poison. It does something to the muscle to cause us to stop exercising. And we may want to, have recovery mechanisms after exercise to flush lactic acid out or prevent the buildup of lactic acid, things like that. But we're actually going to talk about lactic acid being a little bit different, and that lactic acid and the contemporary thinking is it's a fuel. So let's look at it. In this section, there's a focus on lactate production and removal during exercise. We're going to need to talk about production, and we're going to need to talk about removal. Is lactic acid levels, or are lactic acid levels a cause of fatigue? Are people stopping exercise because of lactic acid? Now I'll jump to the end, so we're bookend this presentation here. The take-home message from this section is you have to be able to distinguish between lactate threshold, ABLA, onset of blood lactate accumulation, anaerobic threshold, ventilatory threshold, and lactate turnpoint. And I say distinguish between them. You've got to identify how these are similar terms and yet how they're different terms. And I want you to understand the lactate shuttle. Lactate shuttle, in essence, is that you are shuttling, or the body shuttles, it moves lactic acid from where it's being produced to where it can be used by another muscle and used as a fuel. So it may actually be a mechanism to transfer fuel from one tissue to another, from one muscle fiber to another muscle fiber. And truly, lactic acid may not be as bad as we thought. I think the other thing to keep in mind is that the way we measure lactic acid is we do a finger prick. We prick the finger, we pull a little drop of blood off, we put that blood on a test strip. We put that test strip into a sensor, and that sensor tells us how much lactic acid is in the body or in the blood. What's interesting, if we do that lactic acid for anybody at any time, there will always be lactic acid in the system. It's never zero. So even if you're resting and we do a finger prick, your lactic acid is not zero. It's always something, some resting value. It's only when you start exercising above a certain point do we start to see an accumulation or buildup of lactic acid levels. And that's the part that we want to talk about. Okay, so let's get with it. Energy production during supramaximal exercise. So if you do your graded exercise test and you identify the velocity that you elicit VO2 max, this would be running faster than that velocity or cycling faster than whatever resistance was used to elicit the VO2 max. So these are intensities greater than 100% VO2 max. For example, 100-meter sprint in 10 seconds, the average speed is 10 meters per second, that's 22 miles per hour. If we use the ACSM equation for calculating VO2, that would come to a value of 140 mLs per kg per minute. That's far beyond what we would typically see, that we have ever seen in humans. If we see something around 85 mLs per kg per minute, that would actually be quite a bit. Actually, that 140 is not from ACSM. ACSM equation is listed there, and that'd be 123.5 mLs per kg per minute. So clearly a lot of rate of oxygen consumption. Either way, the VO2 requirement for running that speed, something faster than what we would use to elicit VO2 max, is far greater than any VO2 max ever recorded. So energy is transformed from chemical to mechanical energy in some other way other than oxygen dependent mechanisms. So there are oxygen independent mechanisms. And you know these to be anaerobic metabolic pathways, to produce ATP, which is used in the sliding filament theory, cross-bridge formation. We have anaerobic systems without the need of oxygen, ATP-CP, adenosine triphosphate phosphocreatine, and glycolysis. And then we have aerobic metabolic pathway. That's where we're using oxygen. So I've just started drawing those out right here. PC, we have phosphocreatine and adenosine diphosphate. Those combine to make adenosine triphosphate and creatine. That's anaerobic. Glucose is broken down in the muscle sarcoplasm to pyruvate, and along the way that generates ATP, and a byproduct of this pathway is lactic acid. So I put lactate LA in square brackets to represent blood lactate. Glucose is also broken down to pyruvate in the sarcoplasm, and then if oxygen is available, and actually if NAD is available, pyruvate gets pulled into the mitochondria and it's converted to acetyl-CoA. And then the acetyl-CoA gets converted into another substance in the Krebs cycle. And the Krebs cycle continues. And as the Krebs cycle continues, electrons are pulled off and combined with electron transport. And then those are sent to the electron transport system, and lots of ATP is produced. So in KIN 391, hopefully you learned the steps of the Krebs cycle. For this class, I'm not interested in going through the steps so much as understanding that lots of ATP is produced in the Krebs cycle and electron transport chain, electron transport system, either way. That's where the majority of ATP is produced when breaking down glucose to yield ATP. And that all happens in the mitochondria. Fats are also converted to fatty acid in the mitochondria, inside that dotted line. The fatty acid is converted to acetyl-CoA. It also goes into the Krebs cycle and the electron transport chain, electron transport system. That all happens in the mitochondria. So fats and glucose can be broken down to yield ATP that's used for muscle contraction aerobically. Okay, so we have two systems: anaerobic without the need for oxygen, and aerobic. And glucose is the weird substance in that it can be converted to ATP, or it can be used to generate ATP anaerobically or aerobically. And as long as oxygen is available, as long as NAD is actually available for taking on electrons as this chemical reaction happens, pyruvate would be pulled into the mitochondria. All right, so oxygen-independent metabolic pathways. This is the way Noakes likes to talk about it. And it's a nice way to think about it because oxygen is always present in the human body. We're always breathing. We always have some oxygen circulating within the bloodstream. So there's never an anaerobic situation, never a situation where there's no oxygen available, typically. Okay, maybe we could come up with some, but typically we do not have the lack of oxygen present. So what Noakes tries to draw attention to is we have oxygen-independent metabolic pathways. We have metabolic pathways that do not require oxygen. ATP and phosphocreatine stores, there's enough storage to support about 3 seconds of contraction. So running away from a bear would be an example, or maybe a sprint would be another example. Oxygen-independent metabolic pathways, the fate of glucose. Glucose is broken down to pyruvate. The next stage is conversion to acetyl-CoA. This happens in the mitochondria. If pyruvate is not converted to acetyl-CoA, then pyruvate is transformed into, ultimately, lactic acid. And this happens in the sarcoplasm. The biochemical steps leading up to the creation of pyruvate or lactic acid are the same. And therefore, some references refer to aerobic and anaerobic glycolysis. But again, Noakes emphasizes that glycolysis refers to production of ATP without oxygen, but oxygen is always present. It's just which pathway is used, and it's actually determined by the availability of electron acceptor, NADH. Oxygen-independent glycolysis, the endpoint is lactic acid. So technically, glycolysis is the hydrolysis of a sugar the steps that lead up to pyruvate or to lactic acid, depending on what happens next. Pyruvate is transported into the mitochondria and enters the Krebs cycle. Nicotinamide adenine dinucleotide, NAD, must be available to accept hydrogen ions. That's what I was referring to. If NAD is not available, pyruvate can accept hydrogen ions, and then lactic acid is formed. If NAD is available, pyruvate gets pulled into the mitochondria and converted to acetyl-CoA. The formation of lactic acid happens in the sarcoplasm outside the mitochondria. It requires some energy. There's an energy investment, but more energy is yielded. That is, more ATP is generated, and this happens at a fast rate, faster than aerobic conversion of glucose to ATP. Now, lactic acid is an endpoint of glycolysis. Now, it's an acid, okay? We measure levels of acid by using pH. At normal pH, a normal pH of the body is 7.35. Lactic acid will quickly dissociate to form lactate. So it's a very unstable substance. And so sometimes you'll read lactate, sometimes you read lactic acid. You can use these terms almost synonymously, almost interchangeably. Lactic acid is unstable and is rapidly converted to lactate. Now, muscles produce and use lactate. I know you're all familiar with the production of lactate, but now I'm going to introduce, maybe some of you haven't heard that lactate is used by muscles. The rate of production increases with exercise intensity. The classic view of that is lactic acid was associated with fatigue, cramps, side stitch, post-exercise muscle soreness, and other things. And part of that was because proteins do denaturize in acidic environments. But the acidity is not changing dramatically with the production of lactic acid. The contemporary viewpoint, as I've mentioned, is lactic acid is a fuel, and it's shuttled to muscle fibers or even other tissues that can use or reconvert lactic acid. NAD is available. That lactic acid is then converted to acetyl-CoA and pulled into the mitochondria for aerobic conversion of glucose, ultimately to ATP. So lactate threshold. This is a classic term. It's the point during a graded exercise test where there is a systematic increase in lactic acid concentration. The basic concept of this is that it is a point that signifies when ATP is being generated to a greater extent anaerobically versus aerobically. Therefore, this point is also known as the anaerobic threshold. All right, that's what the anaerobic threshold is defined as, the point where anaerobic metabolism occurs at a greater rate during a graded exercise test. Ventatory threshold is about the same thing, but it's a little different too. This is identifying the anaerobic threshold by measuring gases, and gas being volume of carbon dioxide exhaled and volume of oxygen inhaled. Specifically, the ventilatory threshold is a function of when VCO2 exhaled starts to happen at a faster rate during a graded exercise test. And this corresponds to a greater production of lactic acid, because production of lactic acid, one way to buffer the lactic acid, that's a mechanism to that we call, when we get rid of the acid part of lactic acid, and a byproduct of that buffering mechanism is CO2. So when you start seeing more production of lactic acid, you start seeing more production of CO2, and therefore more CO2 is expired. All right, so onset of blood lactate accumulation, ABLA, is a little different. This does not necessarily coincide with any of these points: lactate threshold, anaerobic threshold, or ventilatory threshold. This point is a fixed level of lactate concentration in the blood. And what you do is you monitor lactate until it reaches some predetermined level of lactate. For example, 4 millimoles or 2.5 millimoles or something different. But the onset of blood lactate accumulation is built around the concept that there is a value that you're looking to exceed or to stay under, okay? In some sense, it's similar to anaerobic threshold in that it's just another way of thinking about when the anaerobic pathway is really accelerating in terms of contribution to generate ATP. All right, so this figure is really important, 3.22, and I want to go through this. There's 4 figures here. I'm going to do one at a time. All right, figure A. On the y-axis is blood lactate levels. On the x-axis is running speed. This is a graded exercise test with intensity increasing over time and increasing over the x-axis. You'll see some different markers here. Right here, 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 here. These are different finger pricks that happen during the test. So during a graded exercise test, if we're doing lactate, we'll grab the person's arm, we'll prick their finger, and we'll try to pull off some blood right then and put that blood drop onto a test strip, and then put that test strip into an analyzer, and then we get the blood lactate levels. It's a little cumbersome, but you can do it. It just takes a lot of practice. And here, we've got 2 different symbols, the big circles, okay, and the little circles. Well, what that is referring to is the number of samples. The test strips are actually a little expensive. Every finger prick would cost about $2.50. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's $25 right there. So it can get a little costly if you're doing multiple subjects. If you've got 10 subjects and you're doing 10 finger pricks, you're already talking $250 of doing that type of study. So sometimes people will do fewer pricks, and maybe they only do a prick here, 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 and here. So maybe only 4 finger pricks. All right, that's important to know because the number of data points that we take in during a test may influence where we actually identify the anaerobic threshold. Okay, so now that I've set the stage here, let's look at this. These are low levels of intensity of exercise. No problem for the person to run these. Blood lactate is existing in the blood. It's present. So low levels, blood lactate low levels of intensity, blood lactate levels remain pretty constant. But at some point, the speed gets such that lactate levels really start to increase incrementally. Okay, that's important. So a classic idea is muscles became progressively more anaerobic, more lactate produced. The point of increase is not always easy to identify and is debatable, and it can be a function of sample rate. So if I only used these 4 data points, I may identify the lactate, the lactate threshold or the anaerobic threshold at this point, whereas maybe it occurs later if I sampled at a higher frequency or earlier. And so sample rate can influence where that anaerobic threshold is determined. Now, the contemporary view of this phenomena of increasing lactic acid, and this happens, we see it all the time when we do a graded exercise test. But the contemporary view is that the amount of lactate that we're seeing at any one of these data points is a function of not only the amount of lactate being produced, but the amount that's being reused, right? That's this part right here. Okay, rates of lactate production and use, 2 different lines. I've got this line right here, line A, same test, by the way, same test, intensity increasing over time. I've got line A here, which represents the lactate production. And then I've got line B, which represents the lactate use. Okay, that represents lactate use, or that says removal. I blocked that out for some reason. Lactate removal. The removal part means lactate was pulled back into the mitochondria, converted to acetyl-CoA, and aerobic metabolism continued, either within the same muscle fiber or that lactate was transferred over to another muscle fiber, and then it was reused for, pulled into the mitochondria and used as fuel. So some of the lactate may never reach the finger prick to where we detect blood lactate. Okay, so lactic acid is used by muscles, the ones producing it, as well as others, maybe the heart, maybe the liver. Lactic acid levels are a result. This is really important. Circle this one. The lactic acid levels are a result of the difference between the amount produced and the amount used. The presence of lactic acid in the blood is thought to be a mechanism to prevent acidic conditions within the muscle cell. There is not necessarily a clearly defined threshold of increase in lactate production. All right, so let's look at this. See this gray area in here? That gray area is the difference between production and use. That's what's going to show up in the blood test, in the finger prick test. So see how this difference right here is consistent? There's no change. That's what this is right here. No change. But as the production starts to happen faster than removal, we start to see a divergence of these two lines, and we see a gap. That gap is what's happening here. This increase that we're seeing in blood lactate is a function of the amount being produced and the amount being removed. Okay? So blood lactate acid concentration, lactic acid entry into the bloodstream minus lactic acid removal. This is the lactate shuttle. And shuttle meaning you're moving lactate from one muscle to another organ or another muscle, and then it's being used as a fuel. So it's never hitting the finger prick. Okay, so that's a really important thing. So now, where does this threshold of production happen? Because clearly there's being more lactates being produced throughout here. That means anaerobic metabolism of glucose is increasing even at these lower stage running speeds. But the difference between the amount being produced and removed is what we're seeing. All right, so let's go to the next graph. Okay, this is just an Alba concept. So this bottom graph is, again, using the finger prick. This is the same data set here, but now you're identifying a predetermined threshold, 2, 3, or 4 millimoles, and you're looking for the point where that lactate in the blood has exceeded these predetermined levels. And then you identify those speeds. And those speeds are thought to be important in terms of training, just like the anaerobic threshold speed. It's just a debate in terms of which number is better to use for training purposes. But that's onset of blood lactate accumulation, not necessarily having anything to do with the sudden rise in lactate. Okay, this is last plot is important lactic acid levels while running at constant speed. So this is blood lactate here. This is time, and then this is a data point for running at 18.75 kilometers per hour, slower 18.1 and 17.25. These are 3 different runs. This is the amount of blood lactate in the system for running that speed. It continued to increase. This is not a graded exercise test now. This is just running a constant speed. So here, the production and running at this speed exceeded removal. And so lactate started to accumulate throughout the run. And so the run ended with about 7 millimoles per liter blood, where it started with resting values of about 1 millimole per blood. But running at 18.1 kilometers per hour, there was no accumulation of blood lactate. That is, the amount being produced was matched by the amount being removed. And then running even slower, actually there was some produced here, and then that production actually, or the amount of lactate in the blood started going down. That means that removal actually exceeded production. Okay, so this is a different way of thinking about lactate, is that the amount of lactate we're seeing in the blood is a function of production and removal. Okay, lactate metabolism. Lactic acid levels do increase with intensity. Noakes uses the term lactate turnpoint to represent exercise intensity at which blood lactate concentrations begin to rise visibly. And they use this turn point because he doesn't like the term threshold, and the threshold implies on-off. But really there is not an on-off. There's just this increase in production and increase in removal, but at some point those two start to diverge from each other. So it's careful, he's careful to note that no threshold exists, even though identifying a lactate turn point suggests that there is a threshold. Lactate levels are a function of production and use, and that's important. That's the critical part of this section. Okay, biochemical explanations for increased lactate. At high intensities, carbohydrate is the main fuel. Glucose is oxidized to pyruvate if NAD is available. If NAD is not available, lactate is produced. Lactate is transported out of the muscle cell, or maybe it's used within the same muscle cell. And the question is that done to prevent acidic buildup in the muscle cell? If so, then lactate is actually benefiting performance. So rather than thinking about lactate being a negative to performance, it may actually be helping performance. Okay, so a different way of thinking about lactate. Lactate turnpoint is the exercise intensity, that is the running speed when we're talking about running, associated with the point where lactate begins to rise visually, and that may be a good predictor of performance. So training has the effect of shifting the lactate turnpoint to a higher intensity, a faster running speed. That's a good thing. Determining the lactate turnpoint is time consuming. It really is, and it's a pain. You get your fingers pricked a lot. It gets a little tiring. It may not actually offer any increased predictability compared to velocity at VO2 max. So we just, we got to weigh some of these measurements with practicality, as well as what new knowledge it actually allows for us. But it does, knowing where lactate starts to accumulate at a fast rate does seem to be an important physiological marker, but understanding the mechanism behind it and why that occurs is important. So Lactate Shuttle is really from George Brooks. He has done a lot of work in this area and has come to some interesting statements. Lactate is an important metabolic fuel. Active muscles produce and use lactate at rest and during exercise. Lactate that is produced as an end product of glycolysis is transported into the mitochondria, oxidized via Krebs cycle, if NAD is available, released into the bloodstream where it can be used by other muscles, or it can be released into the bloodstream where it can be used by the liver, even production of blood glucose. So there's lots of different fates of lactic acid. It's not necessarily just a poison. The lactate turnpoint represents the point where a production of lactate exceeds the use. Lactate shuttle represents the movement of lactate away from active muscles, and does this represent a mechanism of transport of fuel between cells? Just a different way of thinking about the buildup of lactate. So lactate and exhaustion. Well, if lactate is infused in the bloodstream, oddly enough, it has no noticeable effects. It doesn't stop exercise from happening. So if I just take a syringe of lactate and push it into the muscle, it's not going to do anything. It is important to prevent the buildup of hydrogen ions, and the formation of lactate actually helps prevent that. Lactate levels are not necessarily high after prolonged exercise. For example, at the end of a marathon, if we do a finger prick, your lactate levels may not be that high, but yet you're exhausted or fatigued at least. Lactate removal after exercise. Is lactate a cause of muscle soreness? Good question, and we continue to look at this. And there's lots of recovery mechanisms that quantify lactate removals. Well, interestingly, lactate levels can return to normal levels within an hour post-exercise. Muscle soreness can occur long after races, in which the intensity is below the lactate turn point. So even ultra marathons where that intensity may even be 50 or 60% VO2 max, it's not very high. Lactate levels never get very high, but yet you can have lots of soreness. And sprinters are not always sore, but lactate levels can be really high in sprinters. So just some challenges to the way of thinking about lactate and the importance of whether or not removing lactate has any physiological meaning. Okay, summary, lactate turn point. That's the point which lactate starts to increase, lactate threshold, anaerobic threshold, ABLA, ventilatory threshold. Those are terms that you should know and what they're representing. Blood lactic acid levels are a result of the amount of lactate produced and the amount used, either by the same muscle fiber or shuttled to another muscle fiber or another organ, the heart, the brain, liver, what have you. Okay, that's lactate. Thank you.